Welcome everyone to the Bergeron Forum. This is the last Bergeron Forum of the semester. There isn't one next week. That's me screwing up in the scheduling, I'm afraid. So you can do your own, reading assignment next week. I want to begin by thanking Dan Houseman and Mari and Dan for their wonderful assistance all through the semester. It's been a difficult time, trying to Zoom and trying to record and trying to have everything work. And they've really worked hard, and I'm incredibly grateful to all the work that they've done. So thank you, Mari. Thank you, Daniel and Dan. Yeah. Just a couple of announcements. Tonight at 6 o'clock in the C.D. Smith Theatre, and tomorrow there is the informal dance showing, which is always a lot of fun. I was going to announce the environmental studies talk, but it's been cancelled. However, very importantly, on Saturday, the US are playing the Netherlands, and on Sunday, England are playing Senegal in the World Cup. Do not miss these important events. Today, we're delighted to have Geoff Lippa from the Department of Biology, and Geoff is going to talk, as you can see, about an RNA world. And as always, the Bergeron Forum is on the cutting edge because it will involve talk about the vaccines. So thank you, Geoff.
Right. Technology is always a little tricky. So if you're expecting that the world is actually a material world, you are wrong. We do live in an RNA world. And so as Emrys mentioned, my title is We Live in an RNA World. So I'm going to talk about that in a couple of different contexts and exploring the importance of RNA from evolution to vaccines. So trying to provide some principles, some basics before we get into some of these more complex ideas of RNA therapy and RNA vaccines. So I know everybody comes from a variety of backgrounds: biology, chemistry, English, philosophy, right? So I wanted to try to provide a little bit of understanding of what is RNA and what is its function. And I'll talk a little bit later on about this picture here, which is one of the most cutting edge techniques we have for editing DNA. And so it is actually where we see the direction of many different types of therapeutics going for genetic-based diseases. There it goes. So what is RNA? So many of you are probably familiar with the idea of DNA, and you're not far off in comparison to RNA. So RNA stands for ribonucleic acid. It is, in fact, genetic material as well, but it's used in the decoding of DNA. So DNA, if you imagine it as a set of blueprints for the manufacturing of a building or an industry, RNA is going to be useful in providing the interactions between what we know about DNA and genes to the synthesis of what we call proteins or enzymes. So this process of exchanging information from DNA to RNA to proteins is something we'll talk about in just a second. But most particular RNA is considered utilized for protein synthesis, but much of it is also used for regulatory purposes, which we'll talk about. But just looking at the picture on the right here, you can probably see a couple of differences between DNA and RNA. So who can give me one difference that you see in the picture here between DNA and RNA? One difference. Feel free to shout it out. Okay, so that's an interesting observation. You probably see that here, right? DNA has this wonderful alpha helix. RNA has this single strand. Now that's a little tricky, and I wanted someone to spot that because not all RNA is actually single-stranded, especially when we talk about viruses. It does exist as a double strand as well, with the same type of pairing that we see with DNA. Is there another difference you see up here? Okay, somebody said uracil. All right, so if you look up here, I've highlighted that in red. There are specific nucleobases for RNA and DNA. One of the bases for RNA is called uracil. You'll notice that thymine on the left here in DNA is not present in RNA. And that's an important characteristic when it comes to the structural aspects of RNA, which give rise to some of the functions that we're going to be talking about today. And then I heard another person shout out the difference between hydroxyls. And that's a bit getting into the weeds, and I don't want to get into that too much, but that is an important difference, right? And it actually plays a role into the stability of RNA versus DNA. So very good. You all get A's. So there's another basic principle of biology we call the central dogma. And so most of the time you hear about dogma in religious studies, but in the context of the central dogma of biology, this states the transfer of information from genetic material, DNA, is ultimately used for the coding of RNA, which is called messenger RNA more specifically. But there are wonderful non-coding RNAs like ribosomal RNA or rRNA, tRNA or transfer RNA, and these are all important aspects and components in the synthesis of proteins. That's why I have this cartoon up here to signify that process. So here's our DNA chef. He's telling them that they need a protein for 2 tables or table 2. And so our little sous chefs here are messenger RNA, ribosomal RNA, and transfer or tRNA. These are going to play a huge role as we talk about this process of the central dogma and ultimately what we call expression. So when we express ourselves, right, we talk about feelings that we express. This is a little bit different in biology. What we're talking about here is how we go from genetic material to the phenotypes or traits that we see daily, right? When you look at somebody, you see their eye color, you see their hair color, you see the skin color. And so a lot of that comes into play because of the proteins that are expressed, the ones that are emphasizing the structural and physiological changes that we see. And so that's why we talk about gene expression. Now, central dogma says that gene expression occurs from the transcription of DNA into RNA, the translation of RNA into protein. And so transcription and translation are 2 subsets of the larger process of gene expression. Now, the questions that scientists ask themselves once they figure the central dogma out was, can gene expression happen in reverse? So can you take proteins and make RNA from that? Can you make DNA from RNA? And that's an important question. The other aspect is, can RNA replicate or act as a protein or enzyme? Are proteins the only things that have functions, specifically enzymatic functions? And this gets us to the understanding of the RNA world hypothesis. So instead of people understand that DNA, genetic material, is really the basis of life in general, in reality, what we know about RNA is that RNA probably pre-existed DNA. And if it isn't RNA, some form of that particular RNA structure pre-existed DNA. And that's the basics of RNA world hypothesis. So life on Earth began with a simple RNA molecule that could copy itself without help from other molecules. So self-replication is a key characteristic of a living organism, or at least early on. And I have this picture here of one of the early texts in the 1960s by Leslie Orgel. It's called The Origins of Life. And he is one of the foundational or early existing researchers, along with some familiar names that you probably remember from biology in high school. Everybody's favorite, Francis Crick. You might remember Watson and Crick and the DNA structure. So he was working with Francis Crick as well as Carl Waus, who's a microbiologist, in unraveling the functions of RNA. So early on, again, this is the 1970s where this book came out. It was the 1960s when a lot of this research with RNA first developed. But it wasn't until 1986 where the core concept or the naming of the RNA world hypothesis came out. And I've emphasized the idea of hypothesis because this is, again, something that we really can't, because we can't go back to primordial Earth to figure out the basic components and how those components came together sufficiently, we can just conclude that it's a hypothesis. But like all hypotheses, it's based on some data, right? Some basic evidence that we use to determine that hypothesis. So some data that we know about, again, found in the early 80s into the 90s, was that RNA does exist as an enzyme. We call these ribozymes. So they have an interesting name. So we found that RNA can actually self-replicate. RNA can conduct catalysis. It can also conduct 2 other reactions, including protein synthesis. And this is also in the context of what we call the ribosome. So in the cell, there's the ribosome, which assists with the protein synthesis that finishes up the gene expression process. Now, Orgel's book came out, like I said, in the early 70s. A lot of their theories came out in the early 60s. But Orgel did have a very powerful first rule. Whenever a spontaneous process is too slow or too inefficient, a protein will evolve to speed it up or make it more efficient. One modification I'll make to Orgel's first rule is that it doesn't have to be a protein. It could be RNA. And that also presents evidence of the RNA world hypothesis. And we're going to also present RNA as an enzyme, or specifically assisting in these spontaneous processes, these non-spontaneous processes. So that kind of transitions the conversation into the history of RNA therapeutics and technology. So RNA, like I said before, with the concept of evolution, has always been around, but it's also most recently being looked at in the terms of RNA therapy. So what is it? So basic definition of RNA therapy is RNA-based molecules to modulate biological pathways to cure a specific condition. Now, there are a variety of conditions we're talking about here, anywhere from the flu to cancer. And so just to give you a relative timeline for the developments in RNA therapy, 1990s, 1995 specifically, we see the first mRNA vaccine designed to treat cancer, specifically in mice. So when you go and read about, for instance, the COVID-19 vaccines, they have always been here as far as mRNA designed vaccines, right? So this is not something new. 2002, first clinical trial based on RNA interference, which I'll talk about as an RNA therapy in just a second. 2012, this is when we first see the CRISPR-Cas9 editing technology discovered and utilized. However, it was always there in bacteria, so we'll talk about that. And then 2020, as we all know, mRNA vaccine was approved for emergency use for treating COVID-19. Now, I've only listed a few of the developments in RNA therapy. You can see the therapeutics involving RNA are quite ubiquitous. So we have RNA interference that I mentioned, we have mRNA vaccines, we have CRISPR-based editing, but we also have antisense RNAs and RNA aptamers as diagnostic treatments, and then synthetic messenger RNAs, which I'm not going to get into for this talk. I'm going to spend most of my time focusing on RNA interference, CRISPR-based editing, and mRNA vaccines. The other thing that I'll mention is something called a riboswitch. So I mentioned that RNA can exist as an enzyme or have catalysis-like activity. And so a riboswitch is just the same as any kind of switch on your wall or your room at home. So you turn off the switch, turns off the light. You turn it on, turns the light on. And so the way the riboswitch works, again, it's made up of RNA, not necessarily proteins or DNA. So the on version of the riboswitch exists when what's called the ribosome binding site is free to interact with the ribosome for protein synthesis. Now, it's turned off when it binds to a specific ligand. And that ligand in this case is something like a metabolite. So the reason why these exist in a variety of bacteria is because they regulate generally metabolites, the level of metabolites necessary for survival. So this poses a specific antibacterial target. Why would we need a new antibacterial target? Well, it's because of this statement from the CDC and something we see in research. Antimicrobial resistance is rampant. So it is a global public health threat, as it states in the CDC's website. 1.27 million people die worldwide, nearly 5 million deaths in 2019. And this typically comes from bacteria such as P. aeruginosa, Staph aureus. Many of you heard of MRSA, right? Methicillin resistant Staph aureus, tuberculosis, and also we have Neisseria gonorrhea. And so these are typical bacteria that experience antimicrobial resistance, which interestingly enough, they all contain specific riboswitches that could be targeted for antibiotic discovery and testing. So RNA in this regard presents itself as an interesting target for new therapeutic development. So finding, for instance, a ligand analog that could inhibit or turn on or off this particular riboswitch. So what about RNAi treatment? Kind of cut off my top there, but it's okay. So RNAi or RNA interference was first discovered in 1998. So again, we're talking pretty recently here in the context of scientific timeline. Andrew Fire and Craig Mello won the Nobel Prize for RNAi discovery or RNAi therapeutics in 2006. So again, still relatively new. And it was actually 2018 when one of the first RNAi therapeutics came out onto the market. So it got FDA approval, and it's called vutrisiran or Amvuttra, and it's meant to treat something called amyloidosis. So again, you don't know what amyloidosis is, it does not matter. All you need to know is that that's one of the underlying causes of a variety of neuropathological diseases. So whether you're talking about Parkinson's disease or Alzheimer's disease, for instance, those are based in amyloid production. So this is not going to cure those diseases, but it is going to release some of the symptoms associated with them. And so again, RNAi is pretty common nowadays. Is also, as far as how it works, so the idea of interference is key here. Generally, they design a synthetic piece of RNA. That synthetic piece is inserted into the cell, and the siRNA or the RNA synthesized piece will unravel in the context of a large protein complex known as the RISC complex. And now what you've created here is a flag that's going to bring in messenger RNA. And when it brings it in or pairs with the messenger RNA, it will actually lead to cleavage and degradation of the messenger RNA. So you might ask yourself, why is this important? Well, remember, messenger RNA is the connection between DNA and proteins and protein synthesis. So without the messenger RNA, you don't have protein synthesis. So a lot of people talk about this technique or this type of treatment as a knockout type of treatment. So you're knocking out a specific gene or protein that's typically being made. And this is typically used in eukaryotes, in other words, animal species, variety of animal species, as well as you can see it in plants being used too, because they contain this natural or endogenous RISC complex. So besides RNAi and riboswitches, we also have new technologies at our disposal. CRISPR-Cas9 gene editing, also known as just CRISPR gene editing or CRISPR gene therapy, is something that's typically very common nowadays in research. So this particular discovery was awarded the Nobel Prize in 2020. So this is very recent by two female researchers, Emmanuelle Charpentier, which I'll probably totally ruin her French name. I apologize for that. And Dr. Jennifer Doudna. And so, again, this was pretty recent. It was originally found in bacteria, and bacteria, again, for years, millions of years, have used it as an antiviral mechanism, right? To, again, knock out specific viral issues or viral attacks. I'm not going to talk about how exactly that works in that context, but I will talk about how gene editing works in the larger complex. But the idea here is you can now disrupt a specific gene, you can correct a gene, you can remove a gene, or you can add in a new gene. And so that's a lot of versatility for one piece of technology. And so it has been connected to gene therapy because as you add or remove a gene, you might remove a dysfunctional gene, you might add in a gene that's actually functioning correctly. So something like muscular dystrophy, you might remove dystrophin, which is a gene specifically that's not functioning correctly in muscular dystrophy, and put in one that's working correctly. So that's the idea. But there are treatments for HIV, cancer, and also blindness in clinical trials for utilizing CRISPR gene therapy. And this is from 2017 to 2018. Now, it does pose questions about ethical considerations. So CRISPR works best, CRISPR gene editing works best when you're editing an embryo, when you're in the early stages of development and cell multiplication. right, or cell replication. And so obviously, if you're going to do this, what are ultimately going to be the final results? Now you have what you expect to happen when you make those modifications to the DNA. But you also have side effects that you're not going to know about until that particular embryo is turned into a child and ultimately into an adult. So as you might expect, with those updated ethical considerations, there's new FDA guidelines to the use of CRISPR-Cas9 gene editing, but also how it's going to be researched. And one of the reasons why this has come to be the forefront is because of the article you can see here, where we have a Chinese scientist says he made gene editing twins using CRISPR technology. This was back in 2018. So the researcher that's mentioned in this article, gene edited twins, specifically trying to make them resistant to HIV infection. So he changed specifically a receptor, so a flag on the surface of a white blood cell, an immune system cell, so that HIV wouldn't recognize that particular cell and therefore attack the individual. Now, again, we don't know what the long-term effects are going to be for that. He was arrested for 3 years and jailed. So he's just coming out of jail now. So we're very interested to see what else he's going to be doing in terms of research. But again, it does pose the question of who controls this technology and how do we use it? So a little bit of ethics for the philosophers out there. So I wanted to show this video of how this process works. And so it's going to be, we're going to try it and see how it works here. It's not showing it yet. So the video I'm going to show you here shows how the CRISPR-Cas9 system works. This is part of the CRISPR Therapeutics, a small startup biotech company. This was started by one of those Nobel Prize winners.
Today, most medical therapies aim to treat diseases by targeting abnormal proteins associated with a disease. Usually, these abnormal proteins exist because of mutations in specific genes in a patient's DNA. In the past, we have not been able to alter these genes directly to address the root cause of a disease. However, a novel gene editing system known as CRISPR-Cas9 may allow us to modify, delete, or correct specific areas of our DNA in order to treat these diseases. The system is comprised of 2 parts, Cas9, an enzyme that cuts DNA, and a guide RNA whose sequence directs Cas9 to a specific location in the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that can be easily and precisely targeted to a desired site in the DNA. The CRISPR-Cas9 gene editing process begins when the complex recognizes and binds to a short segment of DNA adjacent to the target site. This initiates unwinding of the DNA helix, which allows the guide RNA to pair with a specific target sequence in the DNA. If the sequences pair precisely, Cas9 cuts the DNA, forming a double-strand break. Our cells respond to such breaks by activating natural DNA repair pathways. In some cases, a process called non-homologous end joining results in the addition or deletion of a few base pairs, which disrupts the original DNA sequence and can cause gene inactivation. A larger fragment of DNA can also be removed by using 2 different guide RNAs to target separate sites on either side of the desired deletion. Cleavage occurs at each site, and the repair process joins the separate ends, thereby deleting the intervening sequence. Corrections to DNA can also be made by adding a DNA template along with a Cas9 guide RNA complex. The template is designed with sequences that exactly match the DNA adjacent to the target cut site. Through a process called homology-directed repair, the cell uses the template to repair the break, thereby replacing the faulty DNA sequence or even inserting a new gene.
So again, I think the video on the side is the first to something like this down the line, and it doesn't even get into some of the other aspects that could be used for in the research lab. So of course, I'm sure most of you are here to learn a little bit more about messenger RNA vaccines, right? In the context of maybe even COVID. So how do these work? So interestingly enough, as I mentioned earlier, mRNA vaccines have been developed and tested since the early 1990s. So it's not like we just developed these in the 2020s. So how do they work and how are they designed? So we'll get into the idea of how they're designed in just a second, because it's very complicated, as you might expect. But how do they work? So remember, we talked about messenger RNA as this go-between between DNA and protein synthesis. So when you look at the picture on the right, this is taken from the NIH website. So if you're interested in this and knowing more about how it functions, I encourage you to go to that website. But what happens is there's something called a lipid nanoparticle, and that lipid nanoparticle contains messenger RNA. So the lipid nanoparticle binds to a specific cell, and then you have the vaccine, which is ultimately the messenger RNA portion of the lipid nanoparticle. Is actually entering into the cell. So it's released into the cell cytoplasm. And so say, for instance, this messenger RNA is for the spike protein that many of you heard about probably in the news. And so it has the messenger RNA that can be used with the ribosome for the synthesis of proteins or an amino acid chain. So the spike protein is being synthesized utilizing the directions from the messenger RNA. So why would this be important? Well, the spike protein can then be presented on the outside of the cell, because in the context of this picture, this diagram, this type of cell is specific to antibody production. So it's oftentimes presented on a B cell or a T cell, which is a specific white blood cell type. So the spike protein is on the surface. You have antibodies and other immune cells that can now bind to it. And this encourages the production of antibodies. So the next time you get infected, say, for instance, by the COVID-19 virus, or SARS-CoV-19 virus, you would ultimately have a defensive mechanism to mount. So how is this different from traditional vaccines? Well, traditional vaccines, you usually are getting injected with something like a weakened form of the virus or a part of the virus like a protein. That's going to create the same effect. You're going to start seeing the synthesis of antibodies, and immune cells are going to start recognizing those flags and attack them and destroy them. So again, that's very simplified immunology. There's a lot more complication to that. So that's how they work. And so, as you might expect, when it comes to the design of something like this, there are multiple levels of complexity. So first off is the nanoparticle, the lipid nanoparticle. How is it designed? How is it going to work best to attach to the target cell, but also can hide from the immune system so it doesn't get destroyed before it actually conducts its function? The other is, how do we design the messenger RNA to be most efficient for the production of the spike protein? And then, of course, we want to make sure that the spike protein that we're making is stable enough to produce antibodies or trigger the production of antibodies. And so the production of the COVID vaccine that we know about today is not trivial. There are many people behind the vaccine itself. that are not even listed on the left here. I've only listed a few of the individuals who were a part of the discoveries based on strengthening the effects of the vaccine. So the first one is Robert Malone. So timeline-wise, he conducted the landmark experiment mixing messenger RNA and droplets of fat and found protein production from that in the context of cells. Then we have Carrico and Weissman, who modified the messenger RNA. So they found that typically when you introduce messenger RNA into an organism, it mounts a very strong inflammatory response, which is actually not going to be beneficial into the final successes of the vaccine. So they've made changes to the sequence. So instead of the uracil, right, that we saw earlier in the presentation, they make what's called pseudouridine or pseudouracil. So they modified that to both stabilize the messenger RNA in the context of the drug or the vaccine, but also to prevent some of this inflammatory reaction. And then you have Derek Rossi. Again, many of you probably heard of Moderna in the news. He's one of the co-founders, but he described how modified messenger RNA could be used to transform skin cells into embryonic stem cells and also muscle cells. So he was able to utilize some of the information from Carrico and Weissman for some of this early testing. And then, of course, we have Peter Cullis and Thomas Madden, who pioneered lipid nanoparticles. So the lipid nanoparticle, for instance, you see on the right here, has 4 different types of lipids. Some of these lipids are to stabilize the structure of the nanoparticle. Most of them are, but some of them are also to, as you can see here, will change their structure and their chemical properties in the bloodstream. And so that could be also important in limiting the immune response to the foreign object coming into the body. Then we have, last but not least, Barney Graham, Jason McClellan, and Andrew Ward, who were responsible for the design of the spike protein that adapts to the shape that's more amenable to inducing immunity, right? So I mentioned optimizing the spike protein structure. So they're all, again, part of that design. And so that does not include the probably thousands or hundreds of other individuals in the various companies like Moderna that were a part of this process of development. But you can imagine that even some things like the development of the fatty nanoparticle could be used for a variety of other purposes for therapies as well, including RNAi therapy. And so again, I just wanted to emphasize how complicated it really was in developing this type of vaccine. So what's in the future of the RNA world? So a couple things that we see going on currently are the advent of so many different RNA-based therapies. There's many new developed research centers or treatment centers. So many of you might be familiar with Roswell Park Cancer Center. So there are many different cancer centers out there that specialize in oncology, but there are also new RNA-based specific research centers and treatment centers. One in particular came up in Texas. This is the Houston Methodist Research Center, and they're specifically looking at RNA-based therapies for individuals. With what I just mentioned with the mRNA vaccine, we're also optimizing RNA therapy stability. So making sure it's stable for injection and therapeutic. Delivery is always a difficult concept. Where does the drug go? It'll start off maybe in the bloodstream. Most of these are injected through IV or subcutaneous, so under the skin. But where do they go from there? How do they target specific organs or tissue types? And then immunogenicity. This is basically how your immune system responds to the therapy. That's always a black box. We don't necessarily know until we try it in a variety of model organisms. I mentioned it already, but CRISPR editing systems are changing modern medicine and research. That's not something that pretty obvious, I think, with the capabilities there. Many of us already know and have already probably received booster shots as well as the new vaccine for Omicron. You might wonder why we have that or why is it needed? So if you think about that spike protein we mentioned earlier, the Omicron version of the COVID-19 virus has about 30 different mutations in that one spike protein. So that's one of the reasons why we have such different responses to the various types of or strains of the virus. So that new one is what's called multivalent or trivalent. So it's actually affecting a couple of different subsets or subvariants of the Omicron disease. And of course, RNAi treatments or RNA interference. So I mentioned already one example in 2018 that's on the market through FDA approval. These are 7 different new drugs as well that are RNAi based, and they're in phase 3 clinical trials. So phase 3 is the last phase before FDA approval. So they're currently being tested in humans. The one that I will point out for you, these are again generic names, so you probably won't see them on the market yet, but fitusiran, which I'm hopefully pronouncing right, is an interesting drug because it actually is targeting those individuals with hemophilia A and hemophilia B. These are blood disorders, if you're not familiar with that, or bleeding disorders. So they found that in clinical trials that the fitusiran works to reduce bleeds by 61%, which is pretty significant for a new drug. And this would also be an extended life type of drug versus what these individuals with hemophilia already experience with usually protein injections to help with that clotting aspect. So there are a variety of different new drugs on the market with, again, a variety of different targets. So that's just one of many. So I'll stop there. This is my references in case you are curious about where I'm getting my information. And with that, I'll take any questions. Happy holidays. I had to throw in a little bit of DNA humor in there. But again, I'll open it up to any questions you might have. I'll do my best to answer them. Yeah. Yes. Okay. All right, I will. I'll try. So the question was about the lipids that were attached to the messenger RNA in the lipid nanoparticle. And yes, those are specifically found to be helpful for stabilization or stabilizing the messenger RNA. Yeah. Yes.
It's a question along the same line. I was curious if the lipid that is carrying the mRNA that allows it to go into the cell, if that lipid is the same for each of the different vaccines, basically, or all the different vaccines coming out.
That's a very good question. So again, I'm not the developer associated with the vaccines, but I would imagine because the mRNA, the general principles of the structure of mRNA, the charges associated with the backbone of the RNA, so the chemical compounds are relatively the same, just slightly different sequences, that the lipids themselves would probably still be the same as well. But you do have that versatility with lipid nanoparticles. So if you're not putting messenger RNA in there, you could put a different type of chemical, for instance, that you want to target as a therapeutic. So in that case, you might want to change the lipids a little bit. Any other questions?
I had 5 boosters for COVID, and I got COVID. Why did I get it?
Can you repeat the question? Geoff, repeat the question.
All right. So she stated that she had 5 boosters for COVID, and yet she still got COVID. And so I think this stems from a basic, a little bit of a misunderstanding or misnomer associated with vaccines. So whether we're talking about messenger RNA vaccines or vaccines in general, what it's doing, it's assisting you in preparing your immune system for infection. So when you do get infected, you're not going to have such an extreme response. So you're not going to go to the hospital, for instance, for treatment. Your body can still get infected by the virus, but your immune system will quickly, hopefully, depending on if you're immune compromised or not, will hopefully assist your body in fighting the disease or the virus as quickly as possible. So yes, it is still possible to get infected with COVID, even after the vaccination. or the boosters, because that's just not the point of the vaccine. It's not to prevent infection. Infection will happen. It's just ultimately how that your body responds in the long run.
Geoff, we have a couple of questions from the chat.
Okay.
Danny Gagny asks, have you heard of the zombie virus found in the Siberian permafrost? Can RNA viruses last 48,000 years and be revived?
Well, I don't know much about the Siberian virus that you're talking about, but I think what was interesting when I was doing some of the research for this talk is that typically messenger RNA in the early 80s and 90s, when they were working with it, they hated it. It was the worst to work with. Not stable, especially compared to DNA, easily degradable, easily contaminated. So it took many years to actually stabilize it in the context of the vaccinations that we're referring to. So I doubt that your messenger RNA or your RNA would be stable enough, even in the ice.
Okay, there's a question in the chat from David Marsh. How would you respond to someone being worried that the COVID vaccine could alter their DNA just because it has RNA in it? I tried to explain about the process of transcription, but totally lost them.
Yeah, so that messenger RNA that I was talking about that was in the mRNA vaccine. So that is basically once it gets into your cell and it's being used for translation. So the synthesis of proteins like the spike protein, it's all done. So it does not get integrated. The word integrate is what we use in biology. It does not get integrated into your DNA. Doesn't work that way. You would need specific enzymes that are not associated with humans or eukaryotes that would help with the integration. So there are viruses that will integrate their information into your genome for replication purposes, but this is not something that will do that. It's just the mRNAs, nothing else.
So in relation to this methods in which we use to stabilize mRNA, could you talk about the differences between how we do it in the lab and how cells stabilize mRNA? Like, 'cause it sounds like there's differences that we use in the lab that a cell wouldn't normally use to stabilize its own mRNA.
All right. Well, so in the context of our bodies, so once messenger RNA is made, so transcribed from DNA, it gets modified. So there are a couple ways it gets modified. At each end of the messenger RNA transcript, as we call it, they get the addition of like a cap. So they have a cap at one end and a cap at the other end. That stabilizes the ends that would be free for chemical reactivity. So that's the way our cells do it. Now, when it comes to protein synthesis, your ribosomes are only going to be able to sense those modified messenger RNA transcripts. So that one is actually the one they use in the messenger RNA vaccine is modified for that purpose. But if you're trying to do something in vitro in the lab and you're trying to do transcription in the lab, you won't probably, with the correct enzymes, you would be able to create the caps, but typically you wouldn't be able to. Yeah. So it would just produce the RNA and you would have those reactive ends. But there's a lot more to that question. So yeah, as I say, we can unpack it for a whole semester if you want to.
We're just about out of time. Any further questions? Becky.
About the boosters, Greg and I are fully vaccinated, but we're hearing that there are new mutations that we can look for, and that we're being encouraged to get fully vaccinated. Am I off track?
Repeat the question.
Okay. So again, it's another question related to boosters. So the idea of the booster, even with it, it's not supposed to necessarily target the certain strains of the virus, yet we're still encouraged to get the booster. And so that's an interesting question. And I'm not a medical professional in any way. In fact, you can have a conversation with the person behind you at the end of this if you want to. I'm going to pick on her because she's my wife. But one of the reasons, again, we get vaccines is for communal response or population response. So it's good to have the more vaccinations, the better, because of the fact that you have strength in numbers. That's generally how we propose vaccinations in biology, but also in medical professionals or healthcare. And so there's more to that, I think, and that's why there's generally probably some consensus that it's better to have the booster than not have the booster. But yeah, it's a difficult question. I don't have a really good answer for that.
Okay, well,
Yeah, sure. Yeah, that's true too. Yeah.
okay. Well, thank you again, Geoff.
Sure.
Very informative presentation.